Okay, after with good understanding of how conjugate focal planes work and how they're actually produced by the series of arrangement of the lenses in parallel manner when their principal axis coincide, we know that overlaid images at the subsequent conjugate focal planes will be seen if you place object in two different planes of the conjugate focal plane. Now images will appear as if the object were actually placed in the same plane. With this introduction about the conjugate focal plane, we are now actually ready to go and see what are the different types of the optical elements and how they are arranged in an optical microscope, inside the optical microscope, and how the optical trail in the microscope would look like. So here in this presentation, you can see that there are a source of light here, which is in the form of bulb. Now I'm simplifying this in the form of a bulb and showing a filament here. Now this filament I'm showing deliberately to show that there is a certain structure to the light source. Light source has certain structure which you can see is non-uniform. So if you actually focus this light source on your sample, which is a cell here, if you focus this light source, you are actually going to show and focus a non-uniform image of the light source on your specimen. I'll come to what I mean with that. But before that, let us see how all optical arrangements are done in the microscope and how from the light source light reaches to the parts of the microscope. So here you can see a light source. After the light source, you can see a collector lens. Now this collector lens, as I said before, light source will emit light in all the different directions from here. However, the collector lens's job is to collect the light from here and give it to the condenser lens further on. After this collector lens, there is a field diaphragm. Now this field diaphragm is again a diaphragm that we saw before, which will determine the amount of light that can pass from the collector lens to another diaphragm placed here, which is referred to as condenser diaphragm. So after the light source, there is a collector lens. Between the collector lens and another condenser lens, there are actually two different diaphragms. Now why would we need two different diaphragms? We will come to that also. It will be very clear why we need two different diaphragms and not just one. Now, after the collector lens collect the light and give it to the condenser lens, before that there are two diaphragms which can actually control the amount of light that reaches from the collector lens to the condenser diaphragm, and then condenser diaphragm itself can also determine what amount of light it can relay it further on to the condenser lens. So far, you take a moment and remember that after the collector lens there are two different types which determines the amount of light that will pass further on to the condenser lens. Now the condenser lens itself has a job to illuminate our sample in case of trans-illumination, right? So this condenser lens is going to illuminate your sample with your light, or like this. How do you want to do that is a difference, and that determines your illumination profile. Now, the amount of light which the condenser lens will focus on to the specimen, after that specimen will actually transmit some light. Now, the transmitted light from the specimen is actually collected by another lens which is referred to as objective lens. This specimen which is placed in a particular stage is referred to as specimen plane. And after this specimen plane, the objective lens will be there, which will actually collect the information from our sample. As the light goes from the objective lens through the tube lens to the eyepiece. So there are two more lenses coming in the picture right now. There is another tube lens after the objective lens, and then there is the eyepiece. After the eyepiece, you have your retina or a camera where the image will be formed. Now, after looking at all the parts of the microscope, we can see that a light source collector lens field diaphragm, condenser diaphragm, and the condenser lens itself form the illuminating components of your microscope. Again, your light source, your collector lens, your field diaphragm, your condenser diaphragm, and your condenser lens along with your specimen plane forms the illumination components of the microscope. On the other hand, if you see the components which are just above the specimen plane, which are collecting the information from the specimen, includes objective lens, a tube lens, an eyepiece, and further on to your camera, it forms the image forming components of your microscope. So in a particular microscope, a modern day typical microscope has two different type of components, image forming components and the illumination components. Now there are two sets of component we can see here, the illuminating component and the image formation component. So our job is to align the image forming components with the illuminating components so as to get a perfectly uniform illumination on our sample, and we now know what is the importance of that kind of an illumination. Now what do we mean by alignment again here? As I said before, alignment of the imaging components, image formation components with the illumination component means to adjust them in X, Y and Z direction such that both of these individual components are actually aligned and the image that will be formed will eventually have a uniform illumination. The Z direction here, I talked about the alignment in X, Y and Z. So let's see what are the different kind of planes where you can align your optical components. X and Y is basically the components laterally. You can align here in X and in Y. But if you can align the component in along Z axis also, that means along this principal axis which is moving, along this axis if there is any movement you are making the alignment, that is referred to as along the Z, and along the X and the Y it is referred to as lateral. So it is lateral versus axial alignment. Now if we are clear with that, the Z direction in the axial direction that we are talking about along the principal axis of these components. Hence, by aligning, we are actually trying to move all of these components in different combinations of X, Y and Z so as to get a very optimal harmony of alignment between the illumination components of the microscope with respect to the image formation component of the microscope, and both of these are actually with respect to the specimen plane. Now, eventually that will actually determine whether your sample is perfectly illuminated or not. And we all know what we want here. We want our sample to be looking something like this, which is having a beautiful illumination with great detailing and contrast and sharpness, rather than this unevenly illuminated sample. We don't want that, right? For our sample, nobody wants that. So now here we are very clear as to if you align these components optimally with each other, you are going to get a perfectly illuminated sample, which is shown on the right hand side towards the lower panel. Now, in this next slide, you can see a light source in the form of a bulb, which is showing again a filament. After that, you have a back focal plane here. And this back focal plane is basically of this lens placed here. And after this lens, you have a specimen plane. And on this specimen plane, I have kept a single cell as a specimen here in front of you. Now, light source, which is below the focal plane of the lens, if you're going to illuminate this light source, it will start illuminating the light, right? And this light will actually pass in this direction towards your lens. If your light source is basically placed before the back focal plane here, if you're keeping your light source somewhere here, which is back focal plane of this lens, you are actually going to form an image like this, where the light filament is actually going to be imaged onto your sample. So you can see the samples in the specimen plane, you can actually see the image of your light source. Now, again, in this slide, you can see that there is a light source which is placed before the rear focal plane of the condenser lens. Now, this is a condenser lens which is actually trying to focus a light, which is placed onto the stage. There is a specimen. Now, if your condenser lens tries to focus onto your specimen, an image of your light source, an image of your light source means an image of your filament actually, which is a light source. Now, if it's going to focus an image of the light source, you can see that the light source is actually very non-uniform. So in this case of the sample in a human cell placed on top of a cover slip placed on the sample plane, the real image of the light source or the basically the filament will be formed on the sample plane if the light source and the specimen are in conjugate focal plane. So this plane where your light source is kept and the plane where your specimen plane is are actually conjugate focal plane. In that case, your image or a non-uniform image of your light source will be actually coinciding with your sample. And eventually you are going to see what you are actually going to see as if your light source as well as your specimen is in the same plane. Now the real image of this light source, if it is formed on the sample plane, it means that these two types of light source and the specimen are in conjugate focal plane. Now our light source is extremely non-uniform. I'm again emphasizing it again and again. The image of that non-uniform source will be formed on a sample plane, and this is not what we want from because we are now eventually going to get a non-uniform illumination on our sample. Now in the form of image of the source, that means if not the light source which is illuminating your sample, it is rather the image of your non-uniform light source which is illuminating your sample. Now, in order to solve this problem, and if we don't want the non-uniform illumination of your image on the light or the image of the light source on the sample plane, what can we do about it? We actually have to apply the basics that we learned in the previous slide. We know if you are going to keep this light source actually at the rear focal plane of your condenser lens, it is going to give what? It's going to give you a defocused or a collimated beam of light, right? How it is going to happen? It's going to look, okay, if you are going to get this kind of a non-uniform illumination, you are going to get a sample which is not perfectly illuminated in this area while this area is nicely illuminated. But if you are going to take the light source forward and keep the light source on the focal plane of the condenser lens, you're going to get a nice defocused beam of light or a collimated beam of light for illuminating your sample. Now, in this case, you can see that this entire beam of light is actually illuminating your entire sample completely in uniform manner from every place. Now, this is happening because your light source is placed before the non-uniform illumination was happening because your light source was placed before the rear focal plane of the condenser length. Now, if we take the light source and put it exactly at the position of the rear focal plane of the condenser lens, it will come out as a collimated beam of light or a non-focused beam of light, and it will illuminate your sample. So in this case, I have merely moved the source of light to the rear focal plane of my condenser lens, as a result of which I'm getting a collimated beam of light which will illuminate my sample uniformly. So that is what we wanted, right? A collimated beam of light to illuminate our sample perfectly uniformly, and this is what you get and call as clean illumination or uniform illumination. Now the image you see here, it's perfectly illuminated because the illuminating part is also aligned with respect to the imaging part. However, in this case, you can only see that the condenser lens, light source and the specimen plane, all these regions and parts basically contribute to the illumination parts of the microscope. However, a very important part of actually getting a Kohler illumination is to align the illuminating part with your imaging components of the microscope. However, we are yet to understand as to how we align the imaging part of the microscope here. So till here, we have now seen that how it is to be exactly done and what is to be done. We have to just take our light source to the back focal plane of your condenser lens so that a collimated beam of light can be produced. Now, whether or not your light source can be moved from one place to another, that is another question. So we are going to see that. Now, from this, let's actually see what all are the different parts of your microscope, and of all these again optical elements. We are back to the same slide where we are showing different optical elements. Let us see which elements of all these can be aligned and cannot be. Now why I'm saying that, because see, if you are actually going to keep all of these optical elements to be available to be moved in x, y and z direction, I cannot even imagine how many freedom of movement you will have in terms of arranging your optics every bit in x, y and z. That is going to take you a lot of your time, right? Think about it. This makes sense to keep some part of the microscope optics fixed in their alignment in x, y or in z or in all the possible directions, in all the possible planes, and you keep them fixed because, and you only keep some of the parts of the microscope that are available for you to be aligned in the x, y and z direction. It will take only a small time for you to actually align those limited number of the optics and get your illumination perfect. In order to save your time and in order to save the effort that goes into this process, most of the commercial available systems have actually managed to do that. They've actually fixed the alignment of some components, some crucial components in your microscope, fixed in x, y and z, as a result of which you save your time and effort in aligning the microscope for getting a better and perfect uniform illumination. Now, before we see what all components are actually those, let us see again what all different components are actually there to be for your alignment. Now, light source, collector lens, field diaphragm, condenser diaphragm and condenser lens are actually forming your illumination part. Above the specimen plane where specimen cell is kept, there is an objective lens, a tube lens and an eyepiece which has to be aligned, which is a imaging part. Now the imaging part and the illuminating part, that is the part 1 and the part 2, has to be aligned with each other for the uniform illumination. When I say that the microscope companies which are providing you these commercial microscope have fixed some of the part in terms of their alignment in the microscope, so you cannot move these part to align, that they are already aligned. For making our job simple, this was done. So we can see that the light source, before that I will clear the ink. Okay, so in this we can see that the first element that we have is the light source, and after that we have the collector lens, both of which are actually fixed in their position. So we will put a transparent box around them. Now after these 2 days, inside a case, actually both of these elements are actually inside a packed case, so you even cannot see them, forget about moving them. Now what we have next is the field diaphragm, which can only be adjusted in terms of its opening. Now you know field diaphragm actually has been fixed in x, y and z, fixed, but you can actually modulate the size of this opening of the field diaphragm with respect to it as to how much you can actually open and close. But it cannot be moved in x, y and z direction. Therefore, this is also fixed, but it can be actually modulated in terms of its opening, which actually will determine the amount of light that can pass through the adjustment we do. The condenser diaphragm, which is followed by the field diaphragm, is also actually fixed in its position, but it is fixed such that it is fixed along with the condenser lens. These two parts of the microscope together form a condenser unit. This condenser unit is fixed with each other, relative to each other. That means the distance between the two is actually fixed. The condenser diaphragm is also fixed in its position and we cannot move it, but the condenser lens and the condenser diaphragm and the distance between them is kept always constant and is always fixed, so that the condenser lens can be moved back and forth along the z direction, and we can also move this condenser unit in x, y and z. So what do I mean to say is, this component, two components are referred to as condenser unit, we know, and this condenser unit has a movement along x, y and z as well. So all these three different planes of movement, or the three different freedom of movement, is available for this condenser unit to be done. Because don't forget, it's the most important part of your microscope which is going to illuminate your sample. in case of trans illumination. So the condenser lens can be moved back and forth along the z direction, and we can also move it along the xy direction. And when you move the condenser lens with using a knob of the condenser diaphragm, you can also move along with it because the distance between the two has to be fixed. But you can actually control you can control for this condenser diaphragm the opening. So for both of these, actually the opening can be maintained, and both of these as a collective unit can be moved in the xy and z direction. Now we have next, apart from this condenser lens, in addition to it, we have a specimen plane. And we all know that specimen plane can be moved in xy direction to actually focus on certain part of your sample. Now I'm here showing a sample in the form of a cell, which is a single cell, right? But even usually we don't have a condition. Usually we have a condition where we have a dish like this. There are sample cells here, lying here and there, everywhere in the dish. So basically you have to actually move to different parts of the imaging dish, and you have to image different cells. It's not that you're going to image a single cell. You're going to get different parts, and in order to do that, you need to have the freedom in x and y and z also. In some microscope, you can actually modulate the position of the specimen plane, so actually you can have a freedom there. Now after the specimen plane, if you want to actually move some other part of the imaging plane, you can move the sample in x, y and y direction. Now comes the objective lens. Now we all know that objective lens is fixed in its x and y is fixed. But the freedom of the objective lens in z axis remains, which is basically what you do every time you put a new sample or you change the objective lens. Every time you are actually using a microscope, essentially one has to focus the objective lens with respect to the sample, so that this focus is actually along the axial direction or along the z direction. Therefore, objective lens has a freedom along z direction, and it can be moved actually to focus your beam. Now we cannot actually move it in x and y direction, but after that we have a tube length and eyepiece, both of which, the tube lens as well as the eyepiece, are fixed in their position in terms of x, y and z. I don't think any one of you would have moved the tube lens, and especially eyepiece, although it can be removed from the place, however when it is fixed in its place in the microscope, it can actually not be moved from one place to another. And that's why that its x, y and z of these two optical elements are also fixed. Now comes your retina. Obviously your human eye is movable. You can move it in all the dimensions possible, and you're going to get your image of the specimen here, which should be ideally perfectly illuminated if you align the components which I just told. Now with all of this, let us depict all the movable elements as the light elements, and the one which are not going to be, which we discussed and do not have freedom, we are going to fix them in a particular place. How do we do that? Like this. Okay, we know that the field diaphragm can be adjusted in its opening. Condenser diaphragm can also be adjusted in an opening, but it is fixed in x, y and z, and it only moves with the condenser unit itself, which can have a lateral movement as well as axial movement, which the specimen plane also has a lateral and axial movement. After that, objective lens has a movement in axial direction along the z direction, and after that none of these other part of the microscope are actually movable. With this introduction as to what are different optical elements and how they are arranged in the optical trail of the microscope, and what are movable and what are not, and what elements which are able to move have a freedom in which direction and which plane, I hope this is clear, because we are going to use these alignment, because we are going to use the movement of these optical elements which are able to move, and we are going to align them to get a perfect Kohler illumination or a uniform illumination. Now, in a properly aligned microscope, let's say you have a properly aligned microscope, you have already done the process of alignment. In that properly aligned microscope, you will have two distinct set of conjugate focal plane. We studied what conjugate focal planes are, right? If you have any doubt in that, just go back to the previous video. Before you actually watch this section, just get clear with the concept of conjugate focal planes, because you're going to need it here, extremely well versed. Now, what I'm saying is a properly aligned microscope has two distinct set of conjugate focal planes. We are going to talk about two different types of the conjugate focal planes here. The first set of conjugate focal planes are referred to as illumination conjugate planes or a conjugate illumination planes. And they are referred to as illumination planes because these are the planes in which your light source will be imaged, or the image of the light source, that is the filament, will actually be produced at all of these planes. After you align the microscope, wherever the image of this filament will be produced, we are going to call them as conjugate illumination planes, because in all of these planes, the image of the illumination source is being produced. Now, before actually getting into what all conjugate illumination planes are there, I would want to answer a question here, which is why do we need a separate condenser and collector lens? I framed this question here, right? So let us see which will be a collector lens. Like, just after the light source will be a collector lens. Just after that will be a diaphragm, the collector diaphragm, then there will be a condenser aperture diaphragm, then will be a condenser lens. This will be a collector lens. After that we have a specimen plane, after which we have an objective lens, after which we have the tube lens, then after which we have eyepiece. Now, of all of these planes we are seeing, the question was, why do we need a separate condenser as well as a collector lens? Basically, the job of collector lens is to collect light and give it to condenser. Why that job is important? Anyone can think about it. Just pause the video and try to think about it. Okay, so we need a collector lens to collect the light from the light source and focus it onto the back focal plane of the condenser lens, so that the condenser lens will not actually focus the light. Rather, it will allow the light to pass as a collimated beam of light through the sample to achieve a uniform illumination. What I meant with that is, assume a scenario here which we saw. If your light source is basically here, your light source is not able to move on its own. Therefore, if you actually keep your light source, if your light source position is fixed on a rear, somewhat back to the rear focal plane of your lens, it's going to put an image of your light source on your sample, but that is going to lead a non-uniform illumination, right? So what you want instead is, you want to put your light source onto your back focal plane of the condenser lens, so that you can actually get the uniform illumination in the form of defocused beam of light or a collimated beam of light. How are you going to get that? Let's see. The collector lens and condenser lens together does the job. The collector lens basically takes the light from your light source, and it will focus the light exactly onto your back focal plane of your condenser lens. If this light is being focused on the back focal plane of the condenser lens, you are going to see the image of this filament on the conjugate focal plane, which is the back focal plane of the condenser lens. Now, if you place the image of the first object, that is the image of the filament, on the conjugate focal plane, in subsequent conjugate focal planes, which is, see, all of these lenses are basically the parallel arrangement of these lenses along the principal axis. They are all aligned with respect to the principal axis. So if you are going to get this kind of an arrangement, this image of the filament, which is actually the image of the object produced by the first lens, will be taken as the image, will be taken as the object by the another lens, and since it is present onto the back focal plane of the condenser lens, it will act as a point source, and from there the condenser lens will actually pass it on as a defocused or collimated beam of light, which is what you want. We have already seen that. Now after that, the next plane or the next conjugate plane which will be formed is going to be in between the objective lens and the tube lens. And in between the objective lens and the tube lens, it will be exactly at the back focal plane of the tube lens. The back focal plane of the tube lens is where your objective is again going to focus the image of your filament. Now, since it is again at the back focal plane of your tube lens, what will happen? It will again pass it on to the eyepiece as a collimated beam of light or a defocused beam of light. This parallel space or a parallel beam of space is very important to actually incorporate a filters that you are wanting to incorporate, and all of these filter can be actually placed in this parallel beam of light, and it will not obstruct anything here. After that, an eyepiece is going to actually converge and focus the light which is coming in the form of a collimated beam to a particular point, from where this image is going to be again produced on your retina. And however, the image of the filament is only be going to produced by the eyepiece 1 centimeter, around 1 centimeter away from your eye, before from your eye. Again, what are the different conjugate illumination planes available? The conjugate illumination planes that are available is, the first plane is this, where the object itself is placed. The second plane is basically the plane of the condenser diaphragm, which is the back focal plane of the condenser. Then you have the third plane, which is the rear focal plane of the tube lens, or basically it's a rear focal plane of your objective lens, but it's a back focal, that is basically a back focal plane of your tube lens. So that's the third conjugate illumination plane. And then you have another plane, which is basically the plane outside the eyepiece, which is kept around 1 centimeter outside the eyepiece, and that will be the fourth conjugate illumination plane that you have. Now, let me again summarize this entire process, because it's very important to understand before you actually understand the other set of conjugate planes. Now, again, we know why we need a condenser lens and a collector lens separately. Why do we need that? We need that because a collector lens is going to collect the light from the light from the light source, and is going to focus it onto a particular point of the condenser diaphragm, which is actually fixed at the rear focal plane of your condenser lens, and hence your condenser will now illuminate the sample with the perfect defocused collimated beam of light, which is going to give you a uniform illumination. Now, as a result of which we need these two different lenses. I hope we can acknowledge why do we need a collector lens now, and separately in addition to that, why do we need a condenser lens for focusing or condensing the light on the sample. Now we are ready to see all the conjugate illumination planes we have in the microscope. Since it is the illumination planes, we will not talk about the conjugate imaging planes right now. The first conjugate imaging plane that we are going to see after this is the planes where you are going to see the images. But before that, I hope you all have understood what are conjugate illumination plane from this explanation. Please watch this once again before you actually jump and see conjugate imaging planes. I'll clear all of this and we will move to the next part, which is another set of the conjugate focal planes, which is referred as conjugate image planes. Now, what are conjugate image planes and why are they different from this, right? This will be a question. We will answer it in 1 minute. Now, after talking about illumination planes, let's jump to the conjugate imaging planes. The first conjugate image plane where the image of the light source is formed is the back focal plane of the condenser, right? At all the conjugate image planes, they are referred to as conjugate image planes because we will see what are the planes that will actually get the image of our sample on that. Now, conjugate illumination planes, in all of these planes, basically the image of the source of the light was formed. Therefore, it was called as illumination plane. But in case of conjugate image plane, all of the planes which will actually form the image of your specimen will be referred to as conjugate image planes. Now, the plane in which you actually keep your sample is also the conjugate image plane that you have. So with this diagram, I will now put the rays of the light here and I will show what all conjugate image planes are available. But before jumping into all of those planes, we will understand one by one like we did for the conjugate illumination plane. Now, the sample from the light So let's skip this part for a moment and we will focus on this part first from here on. Just don't focus on this part right now. We will focus here. So what is happening? The image of the sample to produce, the specimen has to give away the light. It has to transmit some light. Now the transmitted light from this specimen is going to be collected by the objective lens. Now if this, in the case of a focus of the objective lens, when your objective lens is focusing on the sample, basically it is on the focal plane of your objective, the back focal plane of your objective. As a result of which, the sample, whatever light it will give, it will pass this light in the form of a collimated beam when it is passing from objective lens to tube lens. Now, after it is passing parallel between the tube lens and the objective lens, which will focus it, after the two planes, there is a place between the eyepiece as well as the tube lens where it can focus. The tube lens will focus this. Hence, all the planes where the image of the sample will be formed are known as conjugate imaging planes. So till now what we have seen, the first conjugate imaging plane is where basically your sample is placed. The second conjugate illumination plane is basically the space between the tube lens and the eyepiece, this one. And this is essentially the back focal plane of your eyepiece or the front focal plane of your tube lens. After which, when an image will be formed, basically this image will be actually shown onto your retina, and after the eyepiece it is passes as a collimated beam of light. So in total, we have these two conjugate illumination plane being formed, image plane being formed. Now we have a conjugate image in the planes of the sample plane itself and the intermediate image plane between the tube length and the eyepiece and on your retina or the camera, therefore where you are actually installing your camera. If let's say it was not your retina and through eyepiece that you are seeing, if you're actually seeing it with a camera, your camera has to be placed in such a way that it is actually on the that its back focal plane is in conjugation with your tube lens, so that you will perfectly get your camera aligned with your optics and then optics of the entire microscope, and you're going to get a very perfect, sharply focused image of your specimen on your camera. And hence, whenever you are installing your camera, you should know where exactly the conjugate imaging plane lies. But there is one more plane near which is the conjugation with the other imaging planes. Now we forgot about it in between here. Let's see what is that. Now we forgot about the imaging plane which is lying somewhere here. We are extending the imaging plane beyond the specimen towards the other side in the illumination region. Here there is another conjugate image plane being produced. Now why would that be? Why would we need that, right? And that is the place where actually your diaphragm is there. Why would you need that? The question to that answer is very interesting. Now if you may remember, we also said that the diaphragm position is fixed and we cannot move it in X, Y and Z, and only open or close it to a different extent so that the light can pass through it, and that is what will determine the extent of light that is passing to illuminate the sample. But you see, it is actually in conjugation with the other imaging planes of the microscope. So it essentially becomes one of the conjugate image plane. Now the image of this, if just go and remember, if it is a conjugate focal plane, a conjugate image plane is a type of conjugate focal plane, right? So if you're going to place any object, let's say this is your object number 2 and this specimen basically was your object 1. If you are going to have a conjugate image plane here, if you keep anything here, you're going to get the overlay of or the image of this object in the subsequent planes, right? Like this and here. Now this is what exactly happens. When you have a diaphragm which is actually placed in the conjugate image plane of this field, diaphragm should also be, the image of the diaphragm should also be formed at all the subsequent conjugate imaging plane. And this also is necessary. It is because if you close the field diaphragm a bit, you will see the edges of the field diaphragm being imaged at the subsequent images in the plane. You will see edges of the field diaphragm in the form of a hexagon coming up like this at all the different conjugate image planes. Now, if we think about it, our sample is being imaged by the objective lens, but the field diaphragm is being imaged by the condenser lens. Now, if our condenser lens is not positioned at the right place, then what will happen? This image of the field diaphragm will not be formed sharply, and the subsequent conjugate imaging planes. Again, your sample is basically, which is placed here, is imaged by the objective lens, but your field diaphragm, which is placed on before the sample, is actually being imaged by the condenser lens. If your position of the condenser lens is not proper, it is not going to image this object number 2, that is the condenser diaphragm, or basically the aperture, the first diaphragm after the collector diaphragm basically. It is not going to image it perfectly and sharply. Therefore, in order to visualize this image of the diaphragm or the object 2 perfectly, you need to align the condenser perfectly. Now, here comes the critical part. If you correctly position the condenser lens, which is responsible for forming the image of the field diaphragm, which itself is an indicative of the correct alignment, right? Because if this conjugate image plane also comes in the right place because of the condenser's position, it will actually give you overlay a sharp image of this diaphragm on all the different planes we have in the form of conjugate image planes. Now, this image of the diaphragm onto all the conjugate image plane is indicative of the correct alignment. In other words, condenser lenses, although it can be moved in X, Y, and Z direction, we did not know how much to move in which direction. Therefore, a sharp image of the field diaphragm is used as an indicative reference for our purpose to correctly align the optic of the condenser lens. Because this condenser lens has a movement in X, Y, and Z, you need some reference as to how will you know whether your condenser lens is correctly aligned or not. You will know it indirectly by using this condenser lens to image the diaphragm, and the subsequent images of this diaphragm basically will be produced on the conjugate image planes of the specimen plane, right? But you might be wondering, why not just fix the condenser lens in a place like this and rest of the illumination optics? Because the answer is very interesting. I will let you think about it, why actually we are not fixing the position of the condenser lens, so that we don't have to deal with the optics again and again when we are actually doing the microscopy. I will let you actually think for this answer by yourself till the end of the video, and I'm going to answer it towards the end of the video. Now, this is something basically a tedious job that you have to do every time you turn on the microscope. So why not actually fix your condenser lens and make your life easy? Hold on for that. We will answer that very quickly towards the end of the video. Okay, so now finally we have reached to a stage where we are actually ready to know, after knowing everything, like to what are conjugate focal planes, what are the different parts of the microscope that can be aligned in X, Y, and Z, and what are their individual freedoms of the movement. After that, we also saw what are the conjugate illumination plane, and then we saw conjugate image planes. After looking at all of this information, I think we are now ready to jump into directly see how actually you have to do a Kohler illumination. Now, at this stage where we are ready to see the actual steps involved in the aligning of optics for your microscope so as to get a uniform illumination by means of a Kohler's method, which is referred to as today as Kohler illumination method, I suggest you all should start this process of alignment with a very low magnification lens that is available with you. Now, why would I say that? Just think about it. The image of the diaphragm that you are going to use as a reference to align the condenser lens is going to be magnified depending upon the magnification of your objective and the eyepiece if you are looking into the eyepiece. But if you are actually not looking into the eyepiece and you're visualizing the image of the diaphragm onto the camera screen, then that magnification will not be accounted. Now, if you are visualizing the image of the condenser diaphragm with high magnification like 100x, you're going to have a trouble with very short movement of the in X, Y, or Z. The short movement of your condenser lens is going to make a huge difference. Therefore, it is always suggested to actually start the Kohler illumination method with the lowest possible magnification lens, and it will be easier for you to align the microscope in that condition. And then when you are done with the alignment with the lower magnification objective, then you can go to a higher magnification objective which you want to use for imaging, and you can tweak it little bit here and there, and you are going to get the alignment done with your higher magnification objective as well. Okay, good to go. Now we understand that we have to start our alignment of the optics for Kohler illumination with the lowest possible magnification available for you, be it 5x or 20x or 40x. Don't jump to directly to 100x. Okay, so let's get started. The very first step in doing the Kohler illumination is to actually focus your sample. How do you focus your sample? You focus your sample by aligning the objective lens using in the Z direction and focus the sample perfectly. Now, when you see that it is an unaligned optics, you will see a non-uniform illumination somewhere like this, where in this part your illumination is not perfect, while the other part your illumination is somewhat good. However, this is not what we want, and that's why we are actually aligning this optics. That's why you are actually viewing this video also. So in this case, you can see that we have a condenser lens to be arranged, which is present here. Now this condenser's XY movement can be controlled with this knobs here, while the Z movement can be controlled with this knob here. Now if you move the controller of the condenser lens along the Z axis, it will move in this and this direction, while if you change the direction of the XY, it is going to actually move in this direction and this direction. Okay, so we are clear with the directions. Now the first step that I told you was to actually align the objective lens to focus on the sample, and this focusing is done in Z direction. This Z focusing is very important to bring your objective in focus, and your sample, when your sample is in correct focus, you can see whether your illumination is perfect or not. If it is perfect, just leave it, don't touch it actually, you just start imaging. However, if your focus is after focusing, you see that your illumination is not perfect, go for the Kohler illumination that we are going to see right now. Now, when you see that your unaligned optics, you will see a non-uniform illumination because your condenser lens is not in the ideal position where it should be, as a result of which you are getting an image something like this, which has some bright areas and some dark areas, and you can actually also see the light source being projected onto your sample, leading to a non-uniform illumination. Now, before actually aligning the condenser lens, you close the diaphragm, the field diaphragm that we talked about. Now, if you actually close the field diaphragm, when you are in the process of closing it, you will see that your light will be cut off something like this from here in this region. Now, further closing it, it will actually give you this. After you close the field diaphragm, you can see the image of the field diaphragm here. The edges of the field diaphragm is visible here, but it is somewhat blurry, right? This image is not sharp of the field diaphragm. You will be able to see a tiny boundary of the field diaphragm in your field of view. Now, when you see the edges of the field diaphragm, align the condenser lens. Only when you see the edges of the field diaphragm in your image, then only you start the alignment of the condenser lens, otherwise don't. If you see the edges of the field diaphragm in your image like this and this, then only you can start the alignment of the condenser Z along the Z axis. First, alignment of the condenser lens will always be in along the Z axis or the axial direction, which is this. Now you take your condenser lens in upward or downward direction to actually focus your image of the field diaphragm. How will you do that and how will it look? Focusing the field diaphragm sharply using the condenser lens would look something like this. Because this is a lens which is producing the image of the field diaphragm on the conjugate imaging plane, we'll be able to see the hexagonal edges of this field diaphragm like this. See, there is a hexagonal shape to it. This hexagonal shape is coming from the opening that is being closed. Basically, you close the field diaphragm, as a result of which the edges is visible in your conjugate imaging planes. Now, after the hexagonal edges of the field diaphragm are actually visible to you, now is the time to actually arrange your condenser lens in X and Y direction. Now you can see that the image of this field diaphragm is shifted somewhere toward the left upper corner. You don't want that. You want this field diaphragm to be actually here somewhere in the center. Now to arrange that in the center, you actually have to use the X and Y movement of your condenser lens, and control with this knobs you can actually control the X and Y direction. And doing that, you actually have to get the image of this field diaphragm in the center somewhere like this. After you have managed to actually align the X and Y of the condenser lens and bring the field diaphragm at the center, the only thing that is left to do and achieve a perfect illumination is to open the field diaphragm completely, and you'll get a beautiful alignment along with the image, something like this. You can be proud of this image after you have done the Kohler illumination, because this image is free of any artifacts which are very prone in the light microscopy. It is also having high sharpness and brightness and contrast, because of which it looks very clear. The detailings of your specimen in this kind of an image will be very clear after you do a Kohler illumination. Now, the only thing you have to do after the XY alignment was to open the field diaphragm. And this would actually lead you to the successful Kohler illumination. Now, at the end of this video, I hope you found it useful, and actually now you can go and align your microscope for Kohler illumination. But I left you with one question before that. And that question was, why do we actually have, and why cannot we actually fix this condenser lens, which is actually the culprit where you have to align it in Z and X and Y direction? And why just not fix it? Go and fix it. Get your ask your microscope manufacturer to fix the lens, and you don't have to do all of this before you imaging. It cannot be done for a simple reason. The answer is that because the illumination optics must be aligned with relative to the image formation optics. So since we are using different kind of objectives in a particular system, sometimes you're going to use a 5x objective, sometimes you're going to use a 20x objective, other times you're going to use a 40x objective, and for live cell imaging of nanoscale structures, you're going to use the 63x or a 100x oil immersion objective. Now, when you're going to actually change the length, dimension and the magnification of your objective, you have to switch back and forth between these different alignment, because every time your objectives alignment is different, or your objective is different, you need to align the illumination optics again and again with your objective lens, which is the major part of your imaging lens. You heard me right. This is not something you do once a month or you can get rid of it. This is something you have to. You cannot also call your microscope specialist or a microscope manufacturer to get this done for you to get an optimal image quality, because actually you change in your lab. Someone might want to do the imaging at 20x and you might want to do another time at 40x. You cannot call up your microscope manufacturer and ask him to do it for you so that you can get a better image. You have to learn this skill and master this skill actually to actually get your Kohler illumination done every time before you start the imaging, and every time you actually change the magnification of your objective and change the objective every time while you are imaging. So watch this video time and again, have a master over this technique and get beautiful images like this what you see. Thanks for watching this video and stay tuned.